All right, rock and roll. Okay, what we're going to look at today is all right. Well, what we're going to look at today is an introduction and start of looking at gene regulation or the regulation of gene expression by the process of transcription. That's where you synthesize the messenger RNA, or other RNA for that matter, off of a DNA template, and then later on we'll have RNA processing, which occurs in eukaryotes but not so much in prokaryotes, and then finally translation. We have 3 different levels to regulate gene expression, and that's very, very important because we're dealing with extremely complex organisms, and I am living proof of what happens when gene expression goes bad. I've got genes that should not be turned on, major transcription factors that should not be activated in these kinds of cells, and that's not doing good things for me and what have you. So they're trying to poison them out of existence. Hopefully that will work. Okay, well, at any rate, let's take a look, first of all, at just quick and dirty BY 101 type stuff at transcription. We've got a strand of DNA here. And these, of course, represent the base, the nucleotides, the bases, and stuff like that. Okay, what we're going to do is enzymes will strip it apart, and one of the 2 strands will be the coding strand. In other words, this is a strand that is actually going to be used that's the strand that's actually going to be used as the template, the guide for the synthesis of an RNA. And let's just suppose the sequence here is A, T, C, G, A, T. Okay, here it is. Now, what's going to happen is an enzyme complex, RNA polymerase, will bind somewhere here. We'll call it RNA pol, that's a common word for short. Binds here. It reads this, is a very large complex set of enzymes. It reads this strand. It moves in a particular direction, reads the strand, and as it moves down, it grabs RNA, the nucleotides that make up RNA, and hooks them in. So we will have a C over here, and a G over here, and an A over here, and a T over here. And as it continues to read down there, and of course this is a RNA molecule. Oops, T, sorry. Oh, not T. Oh, sorry. F. I give myself an F here. Okay, I made a big mistake. All right, no, uracil. Whoops. Duh. Oh, man, blame on the chemo. Okay, no, all right, because one of the things with RNA, RNA, of course, uses uracil in place of thymine. Thymine's exclusive for DNA. Sorry about that screw up. Okay, the polymerase goes down and continues, and now we have our RNA, and then eventually these 2 DNA strands join back together. It's all done by enzymes. Of course, to some extent, it's to be done in a laboratory setting using heat or chemicals to separate the DNA strands. Okay, but anyway, here what we have, that's the basics of transcription. Let's put that magic word down here. So transcription is the process of making an RNA using a strand of DNA as a template, the instructions for making that RNA. Now, important point, those of you that saw with me, I mentioned it before. RNAs are always synthesized off of a DNA template. So all the messenger RNAs, the tRNAs, the ribosomal RNAs, the microRNAs, the L and C RNAs, all the RNAs that we have are transcribed off of genes. You don't make RNAs from scratch because nobody knows how to put them together. It's only the DNA template that gives the direction. So in other words, there is a gene for every single one of the, God knows how many different kinds of RNAs in all our cells. That's still wide open territory. RNA biology is just getting more and more and more important. We'll see that later on. But anyway, okay, so that's the basics of transcription. All right, just in case you forgot your 101 or your genetics or what have you. All right, well, a couple other things about transcription. Let's say, suppose we have a really long strand of DNA. Okay, I'm not even going to put the base pairs here. Okay, 2 strands represent DNA. Okay, we can have a gene here. Here's one gene. And this here could be the coding strand and not this. And we read in this direction. Another gene, you can have the same coding strand, but you can read it the opposite direction. Still, another gene may use the other strand as the coding strand. It varies from gene to gene. So one of the 2 strands is the coding strand, differs from one gene to the next. And what direction you read it in also differs from one gene to the next. And it's sometimes possible that you can have overlapping genes, even in eukaryotes. So this could be a 3rd gene read this way. So that way you can put a lot of genes in a relatively small amount of DNA. Okay, so you can have those kinds of things. You do have some overlapping genes. It's not common in eukaryotes, but it is there on occasion. Okay, so that's a little thing about genes. All right. Okay, well, transcription is performed by a set of enzymes called the RNA polymerases. These are the enzymes. Now, many other enzymes and everybody calls them PALs for RNA polymerase. Okay, now, the RNA polymerases, they are what actually read the template strand, the coding strand of DNA, and they're what actually joins together the nucleotides to make RNA. So it comes down to, that's a pretty tough job. Think what you got to do here. Now, other enzymes have separated the strands of DNA. One, you have to position yourself just right in a position just before the gene. Now, in prokaryotes, the polymerase itself generally often can do that. In eukaryotes, we need a whole battery of other proteins to ensure that the polymerase is properly positioned. More on that. Okay. So, in eukaryotes, the polymerase needs a lot of help to be positioned just at the right place to start transcription. Because you don't want to have an RNA that's got a bunch of excess stuff at the beginning. Okay? It's not good. Okay, so that's one thing. Second thing, this enzyme has to be able to read the appropriate strand of DNA. In other words, effectively know what the nucleotide, what the base sequence is. 3, it has to be able to grab on to nucleotides of RNA, not DNA, but RNA nucleotides with ribose as the sugar and uracil as one of the bases. It's got to grab those. It can't grab other nucleotides like the ones that make up DNA. It has to grab those. It has to position them so you get hydrogen bonds between complementary bases. In other words, like for instance, on DNA A, on RNA U. Over here, C, we have G. You have to have these complementary bases so that they hydrogen bond 2 here, 3 here, and stick together. And the hydrogen bonding ensures accuracy. If it wasn't, and you hear this all the time, all the way from 101, if it wasn't for hydrogen bonds between complementary or opposite bases, these processes simply would not work. Because of hydrogen bonding, when you make an RNA, you make about 1 mistake in a million. Now, give you a perspective on that. Imagine your whole textbook, not this one, the one that you pay an enormous amount of money for. That has roughly about 1 million characters in it. Maybe a little bit more than that. Imagine copying that entire textbook and making only 1 mistake. Pretty impressive. Now, DNA replication has to be more accurate still, so we have proofreading enzymes, kind of like the spell and grammar checker in Microsoft Word and stuff like that, that will catch the occasional error and replace it. In that case, you replace it with the right nucleotide. In that case, you drop the error rate down to roughly 1 in a billion. That's pretty impressive. So at any rate, so now we have to, like I said, we have to do several things. We have to position the bases of RNA right next to the appropriate bases of the DNA template strand so that hydrogen bonds can form. Third thing, now we have to join the nucleotides together. That's a condensation reaction. It's an endergonic energy requiring reaction. So when you come down to this enzyme, RNA polymerases have to be very complex because they have several important jobs to do, and each one of them is different from each other. So not surprising, the polymerases are big, huge enzymes. Polymerases consist of multiple subunits, multiple individual proteins joined together in quaternary structure, each playing particular roles in this. We have the the quaternary structure of prokaryotic polymerase. I don't think we have a really good one of the eukaryotic ones yet. We might. I forgot whether they got that or not. But essentially, you're dealing with something like a dozen or more individual protein subunits. The entire polymerase enzyme complex weighs in at several 100,000. It's a big protein, or protein complex. Now, bacteria, prokaryotes, let's just say bacteria. I'm not even sure if that's true with the archaeans, so I won't say anything about these guys. We have a single one-size-fits-all polymerase. Eukaryotes, on the other hand, with more different kinds of RNAs and regulatory RNAs and stuff like that, we actually have three polymerases, three distinct polymerases. All three of the eukaryotic polymerases share several subunits in common, but they also have unique subunits that are different from one to the other. So for eukaryotes, we have polymerase 2, otherwise known as Pol2, and this makes mostly our messenger RNAs, or at least their precursors, plus a few other kinds of RNAs. Polymerase 1 is focused on the ribosomal RNAs. Polymerase 3 does tRNAs, plus a number of other regulatory RNAs. Now, it turns out we can examine some of these things experimentally because there are certain drugs, in fact, we may use some later in the lab. There's something, a fungal product called actinomycin D. And what it does is it inhibits a common subunit to all three. So it shuts down transcription totally. Shuts down all polymerases. But there are some polymerase inhibitors that are specific for one or the other. So we'll put over here to poison really good. We can use actinomycin. It's a fungal product. Actinomycin D. It shuts all polymerases, all eukaryotic polymerases down. Great way to stop the competition. You're a fungus among us, there's a fungus among us. That kind does chemical warfare on its potential competition. Boy, you shut down transcription, that's not good. Now, polymerase 2, for instance, the best known one is alpha-amanitin. What alpha-amanitin comes from, it's another fungal product, it comes from those nasty, toxic mushrooms that are sometimes called death caps, destroying angel, the Amanites family, especially the worst one, Amanites phalloides. These are the kind of mushrooms that sometimes, every once in a while, you hear somebody in the hospital or getting a liver plant or dying from cooking the wrong mushroom. Matter of fact, there was a case in a nursing home somewhere in the South over the summer where the cook went out and collected some local mushrooms. They didn't know what he was collecting, and several people died from mushroom poisoning. Not good. In other words, unless you're a really expert on that, use store-bought mushrooms, not wild ones. Well, the thing is, one of the things that kills you in this is your alpha-amanitin. It shuts down polymerase 2. No more messenger RNAs, a few other RNA types aren't made. You're toast. The other polymerases do work. And then if that doesn't kill you, they also have another toxin that locks up the microfilament cytoskeleton, freezes it in place. End result, it destroys your liver, and then you either die or get a liver transplant, usually the former. Okay, so yeah, if one of these things don't kill you, the other will. Apparently they even taste pretty good. They don't have any bitter, nasty taste. These things are extremely toxic in milligram quantities. They're lethal. So yeah, okay, but they're good research tools, you know. If you're not interested in poisoning yourself, you can poison your biological specimens and cells and learn things about them. Okay, so there are specific inhibitors for these polymerases. So in eukaryotes, because we have much more complex kind of things, we have 3 distinctly different polymerases that are focused on certain elements and stuff. And not surprising, we'll see in a little bit, that means that you have to have 3 different types or classes of promoters. Promoters are the regions that are the foundation of bonding the proteins you need to start transcription. So polymerase II promoters are different than polymerase I and polymerase III promoters. So that way they attract the right polymerase and not the wrong one. We'll see more on that later. Okay, so that's a little quick and dirty on the polymerases. And that gets us into another section. Now, by the way, I don't know how much of this stuff is review for people. I know people here took genetics.